All right, well, I'm very pleased today to have a special guest from overseas, Cameron Neylon. He received his bachelor's at University of Western Australia, PhD at Australia National University, the Research School of Chemistry in protein chemistry, doing some work on ribosomes. And then he did a postdoc at the University of Bath, Wellcome Trust Fellowship. And in 2001, he became a lecturer in the Department of Chemistry at the University of Southampton, working on protein chemistry. And he has a joint appointment at the Rutherford Appleton Laboratory, a neutron scattering facility. And he's going to tell us about some of that. What makes his visit very special is Cameron is working on Open Notebook Science, which is something that my lab is also working on. It's really a pleasure to have people come and share their experiences and to talk about how we can do science in that way. So, Cameron, please.
Thanks, Jean-Claude, for the introduction. It's a great pleasure to come here and talk about what we've been doing in the area of open notebook science, with Jean-Claude being the person who really kicked this off as an idea, and there was the inspiration for what we're doing in terms of trying to make our work open as well. I titled the talk slightly tongue-in-cheek, Beginner's Guide to Open Science, because not necessarily because I'm trying to downplay what we're doing as unimportant, but because I think we are beginners when it comes to figuring out how to do these things. I'm going to try and make the talk quite interactive. I'm going to show you a bunch of things that are live on the web. That's the whole point of what we do in this area. Some of these things may not work. They may break. That goes with the territory. But if you've got any queries along the way, just please butt in and ask questions if you want to, or I'm quite happy to discuss things as we go along. So where I started in this area was with a problem. I have a group mainly made up of graduate students. They work in a lab and they generate data. That data is collected in lab books. It's collected in computers. Some of these computers are ancient. Some of them are running Windows 3.1 because we can't get the cards to run the instruments. So a lot of people outside chemistry, possibly outside biochemistry as well, possibly still think of this as the way laboratories look. And while in many cases the infrastructure and the benches may be the same as when these pictures were taken, that's not what a modern laboratory actually looks like in any real terms. This is actually a picture of McGill Biochemistry Department in 1921, I think. Your modern laboratories, if you can afford nice furniture, this isn't my laboratory incidentally, this is, I get some pictures ripped off the web in the usual fashion, is full of instruments, lots of computers. Some of these are legacy computers, but some of them are up to date. But there's a lot of separate computers, and that's the core of our problem. We have our data spread out over too many different computers, some of which are networked, some of which are not, many of which we're not allowed to have networked because, again, we're running old versions of operating systems. We can't get automatic virus updates and this kind of thing. The data is almost usually not accessible or indexed in any useful kind of fashion. And what's critical is that the data gets lost as both the computers get retired, whether those computers in a write-up area or computers in the lab, and people leave and you don't have their passwords anymore. All of these things are problems that I'm sure are very familiar to a lot of academics. So that's where we started, and we started with the aim of developing some sort of laboratory system, laboratory notebook system. We're very open-minded about what that might look like that would solve some of these problems. So our objectives, our starting objectives were, and these are more or less in priority order, would have a system that would just actually store the data. One place that is backed up, that is reasonably safe, so I can find it. Students can usually find their own data given a day and a half to pull it out of some computer somewhere, but I want to be able to find it even if I don't know what their email address is anymore. It would be nice, the work I do, a lot of what we do is quite high throughput, or we're developing high throughput systems. It's very nice if we could embed into this a system that would track samples so that we know which sample was what. When you go to the fridge and the label is some obscure number in rather poor handwriting, that we can actually tell what it is. That would be quite nice. I also, as Jean-Claude said, I work in 2 different places. I'll come back to that point a little later, but it's really important for me to be able to actually track what my students are doing. I may not see them for a week or 2 weeks, or I've been here for 10 days now, so I haven't seen them for 2 weeks. So actually being able to keep track of what they're doing, making sure they're on track, making sure things are happening, making sure that any problems are being dealt with is quite important as well. And number 4 is kind of the dream, is where we'd like to be. We'd like all of this data to be in a form that can be processed, possibly not by us, possibly by other people, but really to be able to go back and understand if we got this result and that feeds back to some material that we bought from a supplier 2 years ago, can we link those things together? Is this particular primer, chemical, whatever, bad for whatever reason? And can we figure those things out? Or, as I'll come back to a bit later, can I tell who used up the last of my buffer? Little things like that, yeah, that could be quite useful. So those are kind of our objectives, our starting point. So there are lots of systems out there. I'm going to take you through what we've done. This was built out of a blog-based system. The system we're using has actually been built in-house by my collaborators. It's based on a blog format, and it's fundamentally underlying it's an open source engine which has been modified. So it's not a publicly hosted service. It's not a freely available system. But that means we can make changes to it and adjust the way the system works. I've said something that's kind of jargon speak here, a fully flexible system with arbitrary metadata. Okay, so what this means, and I'll show this to you in a second, the system itself has no idea about semantics. There's no presupposed structure of the data that's going in. Everything, any structure that you put in, you're completely free to do, and that causes problems, but it's also very powerful. There is also, and this is an important point, obviously, if you're going to use something as a notebook, there is a full record of the changes. This is not like traditionally, for some reason, on blogs, publicly available systems, you can actually change the date on a post after the event, and obviously this is totally unacceptable for a lab book. So we have a full record of changes. Changes can be made. It's not terribly easy, and that's deliberately so. But at the moment, we don't have the nice feature that you have in a wiki where you can track back through. That's something that will be developed. But at least the systems are there. So the sites down the bottom are just the URLs for the lab books we have, and I'll show you those in a second. There's also I have another blog where we're discussing how this is progressing. I'll put those links back up at the end as well if anyone wants them. So, what does this thing look like in practice? If I go over here, so this is the lab book that's been set up for a collaborator. So this is a project that hasn't started yet. So this is what it looks like before anything goes into it. Conventional looking blog type of thing. I mean, this looks like possibly something you get out of default version of WordPress or something like that. Fairly conventional looking kind of thing. There's some sections down the side. There's a search box. Unfortunately, that doesn't actually work at the moment, but that's something we'll work towards. If I go to where we started with what we're doing here, I go right back to the beginning. This is a blog, and the reason why we used a blog is because a lab book is essentially a journal. And it makes sense. Your first thought is it's a journal. That's what a lab book is. So we can use it that way. That's fine. And that's what we did. Essentially, it was just a process of typing in some of the stuff that was going on. At this stage, actually, we weren't able to make changes. That's a point I'll come back to. But so these are the very first posts that went in. This is halfway through an experiment as it happens. So the students described an experiment down here, and in this very first experiment here, you'll notice one thing down the bottom here, there are 10 comments. So our first thought was, well, you describe what you're going to do, and then if anything changes, you make comments on it, and if anything happens, make comments on that. So a lot of this is actually about the mechanics of the system as you go down. One thing that comes across quite a lot is that tables are fairly awful, but in fact, we've got some problems down here, obviously. But this is the kind of thing we had. So the results of the post were going into comments. So a couple of things came up fairly quickly. I mean, there's a couple of things we can do here. So the student whose lab book this is was working on 2 different experiments in parallel. So she categorized those as this beta-galactosidase and beta-glucuronidase, just two different enzymes that we were essentially doing the same things to. And here you've got immediately the first step beyond a piece of paper. I can now click on one of these and see all the stuff that relates to that particular project. But something that became quite clear early on is that we've got a lot of text here, and it's very difficult to figure out what's going on, how different posts relate to each other. You see that there are a lot of these numbers around the place, and those are actually references to the student's paper lab book. So the student was still using both in parallel at this point, partly for political and safety reasons, because we were obliged to use paper, and partly because she wasn't comfortable with trusting the system at this stage. So she tended to be writing stuff in the lab book, in the paper lab book, and then transferring it to the notebook. But then she's got a series of references here. This again shows you can put in any type of metadata along the way. But the problem was we didn't know, so if we do an experiment, we want to say we use a material to generate another material, we do something to it. We want to link those two things together. How do we know what was the material that went into an experiment? And so the easy way to do that, we thought, was to have this thing called a sample parent. So this is a metadata that's attached to each post. So if I click on this and go through to that particular post, we have a procedure here. This happens to be a restriction digestion of a piece of DNA. And you can see this is the lab book reference where she's done this thing, and this is a reference, it happens to be on the same page for some reason, but on the same page of her paper lab book was where she made the material that went in. And I think you probably immediately see what the problem here is, that as soon as you've got a reaction where you're putting 2 things in, you're in trouble. So we ended up, you can see as you track down here, we had sample parent 2, sample parent 3. This is just starting to get a little bit ridiculous. If I just show you, I'm not allowed to edit this one. So we have this problem. We want to link products, material that's being used through to the materials they came from, through the materials they're going to. And I don't know, it may seem obvious to you, but it took us a long time to realise this, that if we want to link something to something else, then the easiest way to do that is to put a link in, as in a hypertext link. I certainly have come to realise that in this field that you have often come to a situation that there's something that's blindingly obvious once you've thought of it. So at this point we realised that we'd been actually doing this entirely not in a wrong way, but in a way that wasn't going to help us. So what we did at that stage, what the student did, was actually start it again from scratch, and we reorganised the system. So if I now go into the lab book, which he's using at the moment, so this is an experiment done, is that today or yesterday? I've lost track of what the date is now. It's the 2nd today, isn't it? So obviously she hasn't done anything yet today, but this is right up to date. Actually, probably it's that she's been in the lab in the morning and she hasn't got around to putting it in. She tends to put things into the lab book afterwards. So, so what we have now, I'll just track down the right-hand side again and show a few things. So we've now divided these posts up. So one piece of metadata, actually what I should show you is, I'll just show you what that metadata looks like. Oh, I know what that is. I'm not allowed to edit her posts. Again, there are obvious reasons for that. I'll duck out of this one and actually go into my lab book so I know I can edit things. So if I edit this post, I'm just going to show you what this metadata thing looks like when you're dealing with it. So we have a group that each post falls into. And at the moment I'm using this thing called post type. I'll show you why that's not a terribly good idea in a second. But I can type anything into here. So I can have the category name can be anything, and then what you put what the attribute is can be anything. And you can add as many of these as you like. So I'll just back out of that. Oops. So we have these sections now. So we have materials, stuff we've brought in, basically. Notes are just anything that doesn't fit into any other category, really. Descriptions. Procedure, where we've done something. Product, something we've made. Software issues are software issues, unsurprisingly. And I'll come back to the templates in a second. So what I can do is if I just go into the middle of a process, back out of this, and I'll go to, so this is just a category of posts. So if I run down, I'll go into that post. Okay, so there's no data in there. So I'm not actually operating by my own rules. I haven't actually put the gel image in here, which is a bad thing to do. Nonetheless, this is a gel I ran some time ago now. So basically what I'm doing here is an analysis on a set of samples. Yeah. This is a calibration standard that goes into that analysis, and these are my products that I'm doing analysis on. So where did these things come from? Well, if I click on that, this takes me back to a product post. So this is the post that refers to a material, a tube of something. If I wanted to generate a label for this, I can do that. Because each post has a unique ID number, we automatically have a system for labeling each and everything we make. If this comes, so this is, I could press print, but it'll print on a label printer in Southampton, which is not really going to be much help to anybody. But it would print out this label, yep, which has got the date, the time, it's got a nice long code, and the post number, and then that barcode refers to the code for the number. So I've actually got really quite a powerful system here for identifying samples already built in. I can track back to where I made this. So this is the thing where I've made this product, which I've then gone on to analyze this as a PCR reaction. I've now got, I can click through to all the materials that went into that. So I can click back and forwards, and a machine could do this as well, track back through the entire history of a sample. So what we have again here, we're starting to build up a system that's actually really quite powerful for tracking data, tracking samples. Okay, so I just want to show you one more thing. This is a blog, and the idea is you can put anything in you like. You can type anything you want. The problem is, it's the problem with most wikis, most blogs, we really like to organize things in tables. Tables are a nice way to represent stuff, particularly when you're doing things in parallel. If I show you the code for this post, that's it there. All of that nonsense is what created that nice table. This is not something your average grad student is prepared to do. Figure out how to get this coding working. We also have another problem, is that because you can type anything into these metadata sets, it's actually relatively easy to make a typing mistake. Because we're doing the linking manually, it's relatively easy to make the link wrong. This is why we actually needed to put in an ability to edit the posts, actually, because we were just making too many typos, and that was a complete disaster. So, is anyone here a molecular biologist, by any chance?
Can I ask you a question?
Yep.
The last link you clicked on was for something you put on the gel, and you had the gel results summarised in the table.
Yep.
Do you have the gel on there as well?
I should have.
Oh, no.
But I'm yes. One of the reasons why I find this good is because actually it does force you to raise your standards. It's not just the students. It's also me. I actually keep one of the worst lab books I've ever seen. And this actually...
It's kind of another dimension to your comment about having different things in different computers. The gel is just the same as a spectrum which is on a computer somewhere. But
Absolutely.
It's not in digital form, it's in...
Yep. So if I go back to Jenny's lab book, I'll try to find you an example of a gel. Trouble is that she's now running through the system so fast she doesn't actually run the gels that often. I can show you some pictures. Oh, there you go. There's a gel image. That's the result of the analysis, is a picture. It's not the physical object. That is a JPEG rather than perhaps a higher quality image too. Is another issue. So we also have a lot of what we generate is images, but we're also doing analysis on the enzymes we're generating. So there's Excel files that come out of the product of analysis there as well. So the intention is that any data that we would record should be in there, in whatever its raw form is.
Like you said, it's hard to find a grad student who has the enthusiasm to write the code around the table. I mean, this grad student, you know, for probably very good reasons, didn't want to go to the trouble of making a better gel image. So later on, we were pivoting to this.
The problem there's the gel, not the gel image, actually. And to be fair, that's for what she's done here. That's actually for this type of low melting type gel purification. They don't actually get a lot better than that, to be fair.
Okay,
That's actually not terrible. So actually, the light's not terribly good either, is it?
Yeah, it's better now. You can see it better now.
Yeah, sorry. Yes, there are issues with. I mean, as I said, this is a JPEG, which is a bad way to do it. The reason why the students prefer the JPEGs is because the system automatically displays them. For technical reasons, no one's written the plugin to display the TIFF. No one's imported the plugin to display the TIFF here yet. So
I think the JPEG is a very good way to go.
So if I just, so where was I? I'll get into the right lab book. There's actually an interesting question of whether we should have one lab book for the whole lab or one lab book per person. That starts to create interesting issues as well. We can cross-link, but we can't do what I'm about to show you. So the classic problem with recording data in any form, whether it's a LIMS system, a database, or anything, is actually persuading people it's worth putting any metadata in. People are really tend to be, unless it's very, very easy, there's an issue with that. Additionally, we've got this problem with creating tables. We've got just the problem of doing the linking, and all these things are just a little bit difficult. What we want is to make all of that easy so that people do it automatically. We have an advantage in molecular biology in that a lot of the things we do are quite stereotyped. The procedures we use are very similar each time we do them. There's quite a large set of procedures, but nonetheless, each time you do any one of them, they tend to be about the same thing. So there's an obvious answer to that, which is that you have a template procedure which you work from, and then you just put in the material. But you don't just want to sort of have a template which you need to fill in, even. You can do slightly better than that. If I show you, actually I'll show you the gel. So this is the template. So for that gel post that I showed you in my lab book, or I hadn't put in the image. This is the template that actually, or very similar to the template that made that post. I'll show you what these little codes do in a little while, but basically the templates are exactly the same as a normal post. They just have little place markers in them. And those place markers say what is supposed to go in that place. So up there, up here, the system will look for anything that's been labelled as DNA. The percentage symbol is a wildcard, so it's looking for any post for which the metadata DNA is not empty, because those are the type of things you want to run in gel. The box, unsurprisingly, creates a little box that you can type into. We go down here, we have the procedure so that you know actually what you're supposed to be doing. And then down here, this actually will tell it what other metadata to add to the post when I use that template. So when you do something that is stereotyped, that is a common procedure, which is precise to the ones where you want to track what the flow through things is, it's all handled automatically. So if I then use that template, what it comes up, it just renders that in a slightly different way. So I've got a title up here. I can change the title as appropriate. I can select anything that's in the blog that has the appropriate metadata. It comes up as nice drop-down menus. So let's say we'll run our standard again. And these are some products from an earlier demo, but I'll prop in as plasmid. So probably want about 5 microliters of that, 5 microliters of that, say. I can change these things at the bottom if I want to, but I shouldn't really need to. Pop that in. It then goes into an edit window, so if you want to make any other changes to the thing, you can. But this post has actually now been posted. So if I just open up again, that's come up. Now, there is one problem we haven't dealt with here, which is that I pulled down a product that had been created previously. We're still creating those posts manually. And then we actually have to do some linking backwards and forwards to tie all the ends together. So we still have an automation process that we need to solve. But my feeling is that once we've got that process of generating these product posts sorted out, we actually have a very powerful system here that we've got semi-automated collection of metadata. We've got a system that's relatively easy to use because people don't really need to get down into the level of putting the formatting into posts. And we're making sort of some significant progress towards where what our goals were. So if I go back to, is that the right one? Nope, that's not. So this is kind of the where we are at the moment. We have this one post per item approach, and I think this is actually was a real breakthrough. Doing that really creates an information framework where a lot can come out. The templates are really very important. And I'll come back to this issue that we want to develop this so it's got a web services interface so that other things can link into it. So in terms of what we sort of set out to do, we've actually got a pretty good system at storing and recording the data. I can stand here and I can go back into the system and I can track bits of data. I can pull that data out, and the only person who's really falling down on the job of putting the data in the first place appears to be me. So I've only got myself to blame for that. But actually the students are quite good at this, primarily because they're aware that I might be watching. We have one post per sample. Every sample, everything has a unique ID, so we've got a real chance to track what things are, and also who's been using them. So again, this issue of someone using up the last of the buffer is covered. We can monitor researchers' progress. As I said, I've just flipped back here. If I pop over to my RSS reader, and Peter Murray Rust has been very busy again. No surprises there. Nonetheless, so these are a set of posts that have come up. These are the new ones that Jenny's put in since I last looked, which is quite nice. So she's done a bunch of things. So I can follow this wherever I am, pull this down and see what's going on. So I've actually got really quite a robust system for tracking what's happening. And I'll show you how I aggregate that, a whole set of things in a little while. So where we've not, where am I? No, still the wrong one. So what we've not done, we think this system is, broadly speaking, in a theoretical sense, machine readable. We know we've got a fair bit of work to do on what the best way to organise the metadata in to really make it work, but we've not done any work on really showing whether that's true yet. That's the direction we'd like to go in, but it's really beyond the scope of the grant funding we've currently got to do this. So it's kind of a next stage development to really try and do that seriously. But we've made some progress. So I put open science in the title. I haven't actually talked at all about open science. So what has this got to do with it? I have to admit, when I started this project, I wasn't really thinking about the process of making this readily available. I live in Bath, which is the left-hand drawing pin on that map. I also work at the Rutherford Appleton Laboratory, which is the one at the top right, and at Southampton Chemistry Department. That is a map of the entirety of the UK. That's a triangle that's about 80 miles aside. So I do a bit of commuting. But putting that aside, there's this real serious issue of being able to find out what's going on. Now, I showed you that I can pull that through in an RSS feeder, but there's a problem with getting access into the system. The people at the Rutherford lab run a really quite irritating firewall. And so there was essentially, we couldn't find any way of me getting an authenticated login into the lab book in Southampton from Rutherford. And I just got sick of this. I thought, right, let's just make it wide open. That was easy. I had access to it. So there's the first real benefit of actually making things open. It's not so much that anyone else can get at it, it's that you can get at it. And people tend to skip over that as a benefit, but it's actually really quite an important one. And about the same time, I was beginning to become aware of what other people were doing. I started looking around once we made it open. I saw there's a particularly good video that's of a guy called Deepak, what's his second name? Singh. It is Deepak Singh. I was thinking that can't be right. Deepak Singh doing this amazing 5-minute presentation at a conference in Seattle, which really inspired me. Saw what Jean-Claude was doing here at Drexel and sort of the background information and the community that was really building around making these things available and the potential for doing that. And that got us really excited about what we could do. And that's where we really bought into this open notebook science approach. So there's a lot of benefits here, a lot of potential benefits. There's also a lot of confusion. Open science means more things to more people probably than open access does. So we've adopted this terminology, open notebook science, which was created by Jean-Claude to really try and describe a process where really you do your absolute utmost to try and make everything available, ideally as it happens. I use the term as fast as practicable. You can't make things instantly available, and you can't, in reality, you can never make absolutely everything available because there's always something that doesn't get recorded. There's always some information that's just sort of between the neurons of the people involved. But you do your best to take that as far as you possibly can. That's really the key. So there are lots of potential issues with making your material completely open. The thing that everyone starts as says, isn't someone going to steal your results? Answer, probably no. Realistically, yes, people feel that this has happened to them, but I think in most cases, and speaking as someone who's had been scooped by papers now twice in the last 4 months, most of the time, even when people have done this as a result of becoming aware of results, it's because they were already doing it and they've moved to publish faster. Now, that's a problem, obviously, if you're beaten to publication. In many cases, you possibly would have been beaten to publication anyway. But if someone was really coming in and looking at your results and then going away and being motivated to publish faster, you've got a record of them being there. What's more, you've already disclosed it. You have already reported it. You have a rigid date stamp saying, I've done this. It's not peer reviewed. That's an important distinction to make. But it's out there, so you have reported it. And we've actually got a case of this now in my group where we have disclosed a particular technique for fluorescent labelling of proteins about 3 weeks before the paper that scooped us came out. It's going to be interesting to see how that flows through when we try and publish it. There's a real potential to be embarrassed, real potential to put your foot in it and look really stupid. Flip side of that is if you want to get help from people, you've got to ask. So I think this actually is really what scares people a lot, actually, and it particularly scares the grad students or whatever that you're asking to do this, that someone's going to see what I'm doing. And, you know, we're all stupid, really. There's always someone smarter, and we all make mistakes. So that's a real issue. It does require effort, and it probably requires a bit more effort still than putting together a paper notebook. I suspect the reason it requires a bit more effort is because most of us don't keep terribly good paper notebooks. And that, again, this sort of creates a bit of emphasis to push you to doing better. And I think that's a really important aspect of it. There are potential legal issues. For some interpretations of my employment contract, if someone took a particularly rigid interpretation of my employment contract with the University of Southampton, by doing this I could be in violation of it because I'm supposed to report anything through our technology transfer office just so they can find out whether it's potentially commercially viable. That's not really a major issue in most cases because in practice what happens, you don't send every paper to the technology transfer office for most people in most places. I was talking to someone at Oak Ridge yesterday where they actually do have to do that. Every paper, every poster, every note has to be approved by their technology transfer office, which is a nightmare. One area where I am a little bit more worried is safety information. Now in the UK, it seems to be a little different here. In the UK, safety information is very strongly tied to the lab book. So we weren't putting this in, and arguably that is against, again, you know, this is information on how the experiment is done, so we should have been putting it in. I had a postdoc come in to do a few weeks' work on a project, and she said, well, it's a lab book, it has to have the safety information, so it all went up. And I saw this going up, 15 minutes, half an hour after it happened. I was off-site that day, and I thought, what if someone looks at this, uses it, and manages to injure themselves? This safety information is totally inappropriate for anyone working here. Different regulatory framework, different legal framework, different working approaches. Yet the other side of that, it's very difficult to get good safety information. MSDSs are usually useless because they tell you that salt is a carcinogen and that you should wear full respirator to do anything with it. This is actually a serious problem. I'm not quite sure. And there are other issues. You know, there are ethical and political issues. If you're working on animals, some particular stem cells, these kind of things, you know, you possibly don't want those to be made public. Question then, if you don't want it to be made public, should you be doing the work? There are lots of issues here that are not clear. But there are lots of benefits as well. I think that's where we're going to go. I wanted to talk briefly about the differences between the different groups doing open notebook science. Obviously, there's Jean-Claude's group here using a wiki-based system, which is shown up there on the left. The other person who's really doing this in a really notebook-centric manner is a PhD student called Jeremiah Faith at the University of Boston, and he uses a PDF document that he creates each day. The difference between wikis and blogs, I think, is not per se a central issue. It's just a different way of looking at a database of posts. People think differently about how they interact with wikis and blogs. But I think there's more interesting is the difference in how Jean-Claude's group and my group lay things out. So up here we have, what is that experiment? That's 135. So I think I've got experiment 098 here. So this is an experiment from Useful Chem Wiki. This is the final version. So there's a couple of interesting things that I take from this. This is a version where it's now described in sort of complete finalized approach. This is the experiment after all the analysis has been done, after everything's been explained. And we can go back and look at the history, this being a wiki, obviously. There's a history going back of over several months, I think this actually was done over, where there are various things going on, the experiment going on in the first couple of posts, then some comments between Jean-Claude and Khalid on what these experiments meant and how the interpretation should go. Now, if I pop across here, I've actually tried to replicate some of this in our system in the way I think I would go about it. Now, some obvious differences. One page on useful chem. Dum dum dum. There's the first set of posts. There's a second set of posts. There's a third page of posts. Somewhere down here we actually get to a description of where something happens. Now, you'll see a lot of these are NMRs, so the experiment was based on an NMR time course, so taking a series of analyses over time. And I wanted to separate those out because each piece of data, so you can upload the data, the NMR data for each of those things, and I actually have put those in there, in fact. But to associate a particular product with a particular piece of data, so a computer could tell which one was which, it's necessary, I felt, to create a separate post for each of those NMR samples. So arguably I've made it easier for a machine to pass this, but I've also made it fairly horrible to read. Now we can take all those product posts out and just look at the procedures, that's one option. But I can go back through these and go back to the top policy. Again, because of the way that different systems work, I put this picture, or actually a slightly different picture, at the bottom. But the difference, I think the really central difference between the way Jean-Claude's group work and the way my group is working, or really the one student, in fact, is working, is that we sort of try and log the whole process in the visible final form. It's more like a traditional notebook in the sense that the record of what's happened is immediately available. In Jean-Claude's group's case, what the final product is actually more or less not neatened up, that's the wrong word, but is a finalised description. And to go back through the process, it's not immediately visible. You have to go back through the history. I'm slightly uncomfortable with that, but that's just because I think of a lab book as a log of what's happened. And I think it's a question of what these things are being used for. So the object of Useful Chem Wiki is to provide information so someone can come to that and realise how they should do the experiment. That's less true of our system because they have to track back through a series of things to figure out how they ought to do it, but they can see the process that's happened. And I think what we're finding out is actually that the notion of the notebook, we have a lot of biases and obsessions about notebooks, and properly so, because record keeping is important. But a lot of those things don't matter when you're using an electronic system. And I think we've got to figure out which ones do matter and which ones don't. And different things will matter to different people. So, I'll come back to the right one at some point. Here we go. So, and interestingly, again, in Jeremiah Faith's case, this is much more a traditional written record of a notebook. It's quite different again. There's less obsession with linking backwards and forwards and keeping the connection between things. And a lot of people like this because they feel comfortable with it. It looks like a lab book. It feels like a lab book if you print it out. So I think the differences between Jean-Claude's approach and our approach has largely come from where we started. I started from the perspective of wanting to manage the information. I think Jean-Claude started from the perspective of wanting to make that information available. And that's what's actually driving a lot of the differences, rather than necessarily the difference in the science we're doing. I think for most laboratory research, there are electronic lab notebook systems out there, there are LIMS systems out there. But as soon as you're actually doing research, those systems are going to break. Any sort of structure you try and impose on the data in advance of the experiment, you're almost instantly going to break. It's very important to realise that different viewers of the system have different needs. The supervisor has different needs to the student, who has different needs to the visitor. And we're really trying to work through ways of making those different views available to people. Most people start this with a simple journal type approach. They have some sort of online presence and they just keep a journal. And that's actually quite good. I mean, it has a lot of advantages. You can do text searching and all these kind of things. It also has disadvantages. And there's an issue here of getting people in to do things where they don't, you've got to get them over a barrier. And if there are disadvantages, then they're not always going to see the advantages. And I'm not sure that keeping a simple journal provides enough advantages to keep people involved. I might be wrong about that, but it's something we'll have to see how it evolves. Flexible metadata is critical. If you're going to keep metadata, you need good ways of getting it in there, but you desperately need to be able to play with it and change it at will if it needs to be changed. And I think this method of templating things is a very powerful way of bridging a gap between structured systems where you can impose some structure in advance versus systems where you really just want to be able to type some random stuff in and say, I did this, it made this, it turned green, these kind of things, which don't necessarily fit into any straightforward structure. So where are we going with this? I think I've got my slides in the wrong order. We want to take our system, we want to do a couple of things in terms of usability, but the next big step for us is we want to try and make the system a web service. There are packages out there, they don't necessarily work terribly well at the moment, but there are packages out there that let people manage workflows. So you can grab some stuff from here, put it over there, and then combine it with something. There's mashups, you can call these things all sorts of different things. But you've got to make the system available to a computer to do something with that. So for instance, if we have this is say our workflow engine at the top, this is being described as MySpace for scientists, but it's not quite there yet. But it's a thing that will hold workflows and lets people share workflows, but it will ultimately allow people to execute them as well. So you can imagine, oops, I'll do something else first. I've got to show you one other thing that we're doing, which is what we do when there is a data structure. So we're actually using this online, freely available, web-based data system, lovely graphical system which works, called DabbleDB. It's really worth looking at if you're interested in these things. And we're actually making all of our lab stocks available through a proper database. And you can see so these things are publicly viewable on a public page, and these are accessible by JSON and RSS and various other types of approaches. So if I come back here, which is why that one's come up. So we could set off a workflow. Oh, the arrows have gone. It's a bit disturbing. That's better. So we can set off something that might go to our lab book, then comes back, reports something, maybe a data file, maybe a sequence file of a thing, so that the workflow could then go off to the database or to any other database. This could be some other data store maintained by a facility of some sort where you've gone to do some experiments. And then you might want to combine those sets of data and send them off to query another database. I've shown GenBank here, but it could be any sort of other data analysis, which pumps it back into your workflow engine. And then ultimately, of course, all of this has to be put back into the lab book, because you've got a record of how this thing was run. The problem with these workflow engines is that they're written by computer scientists. Well, that's a problem in and of itself. But the problem is that they don't have this concept that I want to know how this was run, when it was run, and what exact parameters we used to run it. They don't really quite pass that concept at all. So we need to keep a record of instantiations of particular runs of data analysis. That's critical for our perspective. So where are we? I think they're a small group, and really I think in terms of people who are really doing open notebook sciences, as Jean-Claude originally defined it, there are really three groups doing it. There are a variety of other projects that use a variety of other names and a variety of other approaches that have some sort of similarities. But it's a relatively small group of people. I think we're seeing small benefits. We're certainly seeing benefits to the community in terms of an open science community. I think we're seeing less in the way of benefits in terms of driving the science at the moment. But as the community size grows and the connectivity grows between those groups, I think we're going to start to see some real advantages. I think a lot of the tools we have at the moment don't really provide clear wins, clear real advantages that are really going to drive adoption by a wide range of people. I think we're still at a relatively early stage, and I'm certainly a bit hesitant to sort of suggest people start using our system unless they really have a specific need which it can serve, compared to trying to persuade them to make their results more open. If we're going to communicate in this way, we need to think about how we provide sort of tools in the wider sense of how people are going to find it. In chemistry world, there's a series of tags that can describe molecules, and things can be searched on that, and you can pick these things up in Google. That's less true for what we're doing. You might think that biology and biochemistry, it will be very clear. We just link back to the database. The problem is that database doesn't actually refer to what I'm working with, and it doesn't describe what I'm doing. We need to think a little bit about how we can use some of the tools that are already out there and perhaps just do a little bit of tagging. I mean, the whole Web 2.0 thing is really based on just picking something and going with it and seeing whether other people adopt it. So, but I think we need to figure out how to do this in an integrated way. But I think overall, I mean, open science is a great term in terms of its ability to get people excited. I don't think we understand what it means in terms of either the terminology or in terms of really what this means. It really is quite a different approach to put your stuff directly out in the full glare of the 2 or 3 people who might look at it. It is a very different way of thinking, and there are a lot of subtleties that I think, or we're certainly still trying to figure out what they really mean and where they're going. And I think there's also a desperate need here to deal with the problem that you cannot simply, in this day and age, evaluate people on the basis of how many papers they've got in a particular journal. It's not the way we're working anymore. I mean, traditional publishing is broken anyway, but we also need a way of actually trying to figure out what people's contribution is in a much wider sense. So I need to acknowledge a few people, a few very important people. I don't do any of the technical development of the work. I just say, I'd like this. And Andrew Milstead, who's a PhD student in Jeremy Frey's group, goes away and does it sometimes when he has enough time. The development of all of this has essentially been Andrew's work. And he's only 18 months into his PhD, so it's really quite impressive. The people who've used this system and helped to evolve it, my PhD student Jennifer Hale, Wendy Smith is a postdoc that came in for a few weeks on this project, and Joseph Shea is another PhD student who's been involved in recording some stuff along the way. We have some funding from not quite our equivalent of the NIH, but broadly speaking, that kind of thing, to develop this specific system. And also Jeremy Frey has a much bigger eScience. It's got a platform grant which provides a 5-year reasonable long-term funding to develop these things in general. If you want to look at some of these things, then those are the websites, and I think I've probably gone a bit over time, but thank you for your attention.
Questions? I thought when you put up a slide a few slides back, and I can't remember the title, but it was like the flow diagram, actually.
Yep.
Yeah, remember the one before that where you showed an actual example of a flow diagram with some events coming in on the right-hand side and events on the left-hand side, and maybe rejoining at the bottom and so forth. I suppose in a way, that's what I was looking for. The databases, the management systems, or whatever we call them, that you and Jean-Claude are putting together, I've seen, they're kind of, I mean, they're literally a window, okay? But I'm always trying to do like this, look behind the window, to see what's there that I could grab onto. I mean, that workflow, not the workflow per se, but the kind of logic diagram that it exemplifies is really, I think, a great thing, because what you could do is you could click on a part of that and go straight to the thing you wanted. So you can have the gel runs, the compounds that went in another box, the NMRs connected behind those in some way. I mean, even if you only just made the next level visible, I think that's really got potential.
That's certainly where we're trying to get to with the idea of making it available as a web service, and it links also to this concept that different people want to view things in different ways. Let me show you one or two examples of that, one of which I forgot to do on the way through. So this is a very simple example, but basically there are 5 blogs operating in my group, and I post to those as well. So I don't want to look at those in 5 different places, and I don't want to look at the stuff that I've already seen. So there are some really quite cool tools out there. So this is Yahoo Pipes for anyone who hasn't seen it. Basically, this allows you. It's essentially meant to manipulate RSS feeds. It's essentially a way of doing graphical programming for mashups. This is simple enough that I can use it, though I have been known to swear at the screen when I can't figure out how to make something work. But what this is doing, and this was wired up in 5 minutes or less, is it just takes all of the feeds from the lab that I'm interested in, it aggregates them into one, and then filters out anything I've written. Relatively simple example, but nonetheless, it shows you the type of thing that we're working towards. If I show you just one other thing, which I think you might find, these are examples. They're not quite what you're looking for, but they are quite nice examples. This wasn't working this morning. I hope it's been fixed. So there's more than one way to look at a lab book. There's certainly more than one way to look at an electronic lab book. And was working this morning. Here we go, that's all right. So one way you might want to look at it is what's happened over the last sort of period of time. And so this is being generated by, this is a viewing product which is freely available again, developed by a group at MIT. And what this does is it takes XML data with some time information in it and generates a timeline. So this is that timeline. Posts are categorized, colored by their category, by their section. So the red ones are products, the green ones are procedures. You notice, so some things, the student here always generates the products first and then adds the procedures. Which is interesting. One of the little issues that comes up and how you think about how these things work. But we can either track back on a large scale at the bottom here. The screen size isn't brilliant here, I'm afraid, but either track back on a large scale or get more into the detail by tracking slowly here. What we'd love to be able to do is impose on this the links, or to have the network of posts to understand how things are linked in together. No one seems to have quite generated the visualization tools that'll do that for us. Yes, Boyd. I'm going to Boston on Sunday, talking to the people there who developed this and have done some similar stuff, and hopefully we'll see what we can put together there. But yeah, absolutely, we need ways of getting in there and letting people manipulate the data in whatever way they want, because that's where the real power is going to come out of the system. We're working on it.
I have a question about your data. Is that data available for different databases like Sci-Fi and Scholar, or Chemical Abstracts, or some other databases?
It's available to them if they wanted to do something with it. This is so the principle we generally work on here.
Just indexing, because if scientists are doing experiments and if they are not referring to your website, they are not using that information for their additional further research.
So there are two approaches to this. Jean-Claude's looked at how people come to useful chem, and what sort of Google searches bring people there. We haven't done that, partly because we don't have the right tracking things set up, which is.
So you indexed on Google?
We are indexed, yeah.
Okay.
So people can.
Google, I can find this.
Yep. So.
Google Scholar.
Not Google Scholar. So this is a discussion. This is a mini discussion. So this is part of this whole cultural shift. Google Scholar is about peer-reviewed literature, so I just did a search on sortes. Then we're not at the top of the list. I hope I'm going to be able to, having said we're going to find it. If I'd done a search on sortes cloning, which is the title of the blog, it would have been right at the top. Oh, there we are. So it's second page. If I was working on a really hot cancer gene, we'd be down the bottom somewhere, obviously. We're not heavily linked to from outside. This is a moderately obscure system, so we're quite close to the top. It's this issue of tagging, though. How do we expose the right sort of data so that people doing searches do find us. And I don't have the answers to that. For chemistry, as I say, if you put in an Inchi key or a Smiles code, or perhaps exposing CML, then there's clear ways of indexing this molecule, and we should put that data in where we are using specific chemicals. But how you do that for a protein or a small DNA sequence, or that's a harder thing that I haven't got the answer to yet.
If you want to go to total electronic lab notebook, writing everything electronically, how do you handle when the students are learning the technique? When they're not doing anything new, it's not the real experiment yet, but it is what they're doing, and they need to show how long it takes, and you need to go over it with them to see what they might be doing wrong if they can't duplicate a well-known experiment or whatever.
It should be in the lab book. It should go in. There's no question of that, and as should experiments that don't work.
So do you?
We go. I'll show.
Do you shove that off to the side in some way?
No. So perhaps the other way to answer the question is: do we promote the exciting stuff up to a higher level? Yes. So we have some ideas about how to do that. One of the things that has become very clear is that if you don't learn a lot by looking at most people's lab books. You need to either find the specific thing you're looking for, or you need some sort of summary of what's going on. What I've been doing personally is I have another blog, which is a completely separate conventional blog, where I try and tag some of the interesting stuff that comes up. I haven't done a great deal of that either, I have to say. Again, Jean-Claude points up stuff on his blog as opposed to the Useful Chem Wiki, where interesting stuff has come up, or where he's introducing an experiment that's going to start up. So there's a series of different levels, and it's part of this view. How do we go from this, which is basically the equivalent of scribbles in a paper notebook, to something which is not a paper perhaps, but maybe a report? So can we automatically generate a report that covers the things you're interested in? And how we go about doing that is a very interesting question. The first step we want to take, and again we don't really have the resources to do it within the current grant, is that we want to somehow create a button where we can say, right, well, we've written this paper, it's going to be published, and I want to generate a supplementary information that has readable all the raw data in it, and whack it out as a PDF.
I think that it's very appealing, not push it off to the side, but how do you highlight the stuff that's an advance, you might say?
Yeah, well, again, I think it's still on the first page here. The student's been struggling for a very long time. Where's it gone? Student's been struggling for a very long time with the experiment she's trying to do. It's a moderately complicated experiment. And I think it's down here. I can't find it. She's actually just got it to work at the beginning of this week. She suddenly multiplied by 10 the number of transformers number of colonies she has in a plate. That's really important to the experiment. And she says that, but I wouldn't have noticed it unless I'd actually read it through in detail. So it needs somehow. I mean, we could just have a piece of metadata that says "worked?" ? Yes. Or this is a good result, or this is something that you want to see. This person wants to see. Yeah, we don't know how to do it yet.
Can you put some kind of tag on?
Absolutely, yeah.
Or there's something where you could assign an experiment a certain number value level or something. Because I think it's a wonderful example that you've got something you're trying to do. The students try 27 times to do this. And 3 years from now, what you want to be able to do is have a new student group go back to attempt number 27 and not bother with 24.
And the reason why it worked was because she used a different brand of tubes.
Is that true?
Yep.
Is it not just...
No, it's absolutely true. She's been struggling because if you know anything about getting DNA into cells, into bacterial cells, we do what's called a heat shock. So you basically heat the cells for a very specified amount of time to a very specified temperature. And the width of the plastic and the shape of the plastic tube makes a huge difference. And I simply hadn't realised they were using different tubes. So last week, before I went away, I said, you need to get some of these tubes and test it out. I said, it's not going to make any difference. So these things happen. And I could have told her that had I known they weren't doing it, but it's a classic thing. She wasn't writing down the brand of tubes she was using. Why would you? Unless you realised it was important.
But I mean, it really does highlight the fact that you have to have different vehicles for different audiences. So someone will not read this lab notebook, they don't read our lab notebook page after page. They'll do a Google search and they're looking for the boiling point of something, and we happen to done the distillation, and so they can follow that. So you need to have another vehicle, like I use a blog to summarize important things, so even people who are not in my field can actually get some appreciation. And then those link to specific experiments. So that does require effort, and you have to decide.
And again, it's this issue that my students are terrible at writing, at noting what's interesting, what's worked, what the context is. They're really actually quite bad at that. And I try and encourage them to do it, but it's one of these things that requires extra effort, because we've traditionally not kept very good lab books because no one looks at them.
Compared to what you first started talking about with getting instrument data, getting spectra of instruments, it seems to me that one of the big problems is the format of how you save that data. And there certainly are some older formats like the JCAM, we were talking about this before. Yep. And there's been some attempts at coming up with XML-based formats that sometimes have problems that are humongous, when you take a spectrum and put it into that format. Do you have any wisdom about, where those kind of things are going? Is there? It seems to me like that's a big sticking point at the moment.
We don't have a problem with storage space, I have to say. So that's not it's not come up as a problem yet. This system will upload a file, any file. It will start to choke if you try and put in something that's 5 gigabytes, but it'll cope with 20 megabyte TIFFs, for instance. I've done that. So it will take anything. What you can then do with that is another question. But you can, so again, to me, this is partly the philosophy of plugins to start with. Someone can write a plugin to display or do something with it. Particularly imagine this for DNA sequences, that you automatically have a little thing that pops up and gives you a semi-editable graphic picture of a plasmid sequence or something like that, for instance. But equally, as part of this idea of making it accessible to the web, that we don't worry about the data format. Someone who's interested in using it has something that comes and grabs the data, and all right, maybe they have to send it off to a translator to translate it into the appropriate format for their thing. But a lot of these things are available as web services now. And it's relatively easy if you've got a package sitting on your computer to make it either look like a web service or make it available as a web service, even if it's just to you. So there are ways and means of sort of pulling these things through. Having just spent 3 days talking about file formats, I don't have an answer. But it seems to me XML is a good thing in that at least it's partially self-describing. So you've got some internal information in there, and then it should be relatively easy for people to put together style sheets and schema that can tell people that I want to do this with it. So that does seem to me to be the way forward, within reason. A lot of the time, what you really want is a couple of columns of ASCII. That's the easiest file format in. Then you argue about what the columns are.
Well, thank you very much, man. Appreciate it.